In this video, we will discuss about this isoferulic acid that triggers apoptosis in the pancreatic cancer cells. So how this isoferulic acid comes to the mind of researcher. This isoferulic acid is basically derived from, is a derivative of ceramic acid which has the anti-cancer properties. However, how this isoferulic acid affect the pancreatic cancer cells, which is the king of cancer, is unknown. So they wanted to check whether this isoferulic acid has some anti-cancer effect or some negative impact on the pancreatic cancer cells or not. So in order to check that, they started with these two different PDAC cell lines, that is pancreatic adenocarcinoma cell lines, this PANC1 and BXPC3. So what I did is they first did this dose dependent CCK assay or indirect apoptosis assay, and with the dose dependent increase in the isoferulic acid, and they are able to see that with increase in the isoferulic acid, the inhibitions of the cells increases. And here the quantifications on the right hand side, where we can see that at higher isoferulic acid the inhibitions ratios are higher, and the similar results are shown for the BXPC3, where also we can see that with increase in the dose of the isoferulic acid, the inhibitions ratios are also high. And once the indirect measurement of the apoptosis gives a positive indications, they wanted to check the apoptosis using tunnel assay. So what I did is they use proponium iodide and annexin 5 with FITC conjugations, and what they are able to see is that when the treatment with this isoferulic acid are done to the control at a lower level, they see a low increase in the cells in the annexin positive and proponium iodide negative, and annexin positive and proponium iodide positive. But this increase, but this positive cells in both the conditions increases when they increase the dose of this isoferulic acid. That is, in the case of high isoferulic acid in PANC1 cell, they are able to see that the proponium iodide positive and annexin 5 positive cells increased from 5.59 to 8.79, but the increase in the annexin 5 positive and proponium iodide negative cells is from 6.78 to 20. So this indicates that is the annexin 5 or this apoptotic cells has a positive relationship with this isoferulic acid, and isoferulic acids can trigger apoptosis in the pancreatic adenocarcinoma cells. And they did the quantifications of the apoptosis rate are shown on the right hand side, where we can see that results are similar. With increase in the isoferulic acid, they see a increase in the apoptosis rate. And when they did the same studies with the BXPC3 cells, they are able to see a similar result, that is, compared to the control, when they induce low isoferulic acid, they see a low increase in the dead cells, that is from 4.05 to 7.42. And but when they use the high dose of the isoferulic acid, the increase is significantly higher, that is 11.62 to 20.83, almost double. So here also we have seen the similar results that are observed in the PANC1 cells, and the quantifications are in the right hand side, where we can see that with increase in the dose of isoferulic acid, the apoptosis rate increases. So both of these experiments, that is, first the indirect experiment, which is based on CCK8 assay or cell counting kit, and another one is this apoptosis measurement using tunnel assay, both of which gives indications that isoferulic acid also has anti-cancer properties, and basically it will trigger apoptosis in the pancreatic adenocarcinoma cells. So yeah, I hope you understood the final results of their experiments. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.